Okay, great. Okay, hello everyone, and thanks for tuning in. So today we are going to have a talk regarding DNA-encoded libraries and how we can implement this technology into antibiotic drug discovery. So to begin with, most of us are part of the center, so I don't have to mention that many things about how important antibiotics are and antimicrobial resistance, which is a multifactorial problem. And today we are mostly going to talk about the lack of new antibiotics and ways to develop. So first thing what I have to talk about is the hit discovery process, which really starts with target identification and validation, hit identification, hit to lead, lead optimization, and preclinical studies. And my area of research is focused on hit identification, which can be done with in different ways, like unbiased screen, focus screen, or even in silico using computational methods. The most common way that this is done in pharmaceutical companies is high throughput screening. And with high throughput screening, companies get huge libraries of compounds, millions to billions, and they screen them against a target. The main disadvantage with this method, especially for an academic setup, is that we need to have a big storage area in order to keep all these billions of compounds. And usually it is done with the whole screening process is done with robotic equipment. So this is something we definitely cannot afford. And also, for every compound that you want to screen against the target, you need to do one experiment. So if you want to screen billion of compounds, you need to do a billion of experiments. And this is kind of too much for an academic lab. So we need to find other methods that we can use that are less time consuming in order to find hits that are good starting points to continue the development from that point. So what we can do is to use DNA-encoded libraries, which is a technology that allows you to use a DNA tag, which is both a reaction manifold and an information carrier. And by using combinatorial chemistry and specifically split and pool synthesis, we can synthesize libraries of millions to billions of compounds in a matter of weeks. And the big advantage of this technology is that we can store the entire library in just one Eppendorf tube. And then the second advantage of the technology is how we do the screening. So the affinity-based selection can happen in just one experiment because the whole library is pulled together and its molecule, small molecule in this library is encoded by a specific DNA sequence. So by using this to our advantage, we can do the screening in one experiment with immobilized target protein, and then we can just extract the binders, and through amplification and high throughput sequencing, we can identify the small molecules that bind to our target. So before continuing to get a bit deeper, I would like to go through some terminology that you will probably hear me using quite a lot for the rest of the presentation. And this is the building block. So building blocks are chemical compounds that we are going to use, but they are going to be combined together and covalently connected to the DNA. And depending on how many functional groups that can act as reaction handles, they can be classified as mono, bi, or trifunctional. And for example, an example of a bifunctional building block is an amino acid that has both an amine group and a carboxylic acid group that can be extended both ways. And of course, these building blocks that we can use can be either purchased by commercial vendors or synthesized in the labs if we want something specific or if we want to keep the cost low. So next thing when we synthesizer library that we have to think about is the chemistry that we are going to use. So since all this chemistry is happening in the presence of the DNA, unfortunately, the arsenal of the reaction we can use is quite limited compared to all the reactions we can use when we do small molecule chemistry. And something we definitely need to have in mind is that the DNA is a polyionic macromolecule, and this means that it doesn't like organic solvents that much. So we are having definitely a solubility issue. That's why we mostly have to use aqueous environment, usually mixtures of various buffers with organic solvents. And I would say that we need at least 40% of water-based solvent in our reactions. So reactions that we usually do with small molecules that are not compatible with water are kind of out of the question for this technology. So this limits the scope a little bit. We also need to use mild reagents, and this is happening because the DNA, as I said before, acts as an information carrier, and we need to keep the DNA sequencing intact during the whole process. Because if we destroy the DNA with strong reagents, then we won't be able to sequencing, so this leads to not being able to identify the hits. So mild reagents, so let's say strong oxidants or acids are a big no for this technology because they are going to destroy the DNA tags. Also, during the process of the synthesis, we don't do any classical purification in between the steps like chromatography. So we need to make sure that the reactions we are going to use are going to give high yields. And I would say a threshold of around 80 to 90% is something we are mostly looking for when we try to optimize the reaction conditions. And also, last but not least, we would like the conditions that we are using to be selective towards the substrates that we use and also have a broad substrate scope. So getting a little bit deeper in the synthesis, let me see if I can have a pointer. Great. So the synthesis starts with something we call the DNA headpiece, which is DNA sequencing, which through a linker is connected to an amino group, for example. We want something that can act as a reaction handle. So at this point, we can extend it and build the small molecule. So this is the DNA headpiece, and we split it in as many wells as the building blocks we would like to introduce during the first cycle. And then we start with the first chemical reaction. We introduce the first building block. And then during the second step, for each building block, we need to do a DNA ligation on the other part of the conjugate. And we introduce a different code for each building block we use. And then we pull all these conjugates together, and we are getting a mixture. And this is how the first cycle ends. So this is an example of having two-cycle library. Usually we go up to 3 because the more cycles you add, the more things can happen if you don't have reactions that give high yield, and this can lead to problems by the end. So for the next cycle, we take this mixture of compounds of DNA, small molecule conjugates, and we split them again in the same number of wells as the building blocks we would like to introduce during the second cycle. So by doing that, in each of these wells, we are having a population of these conjugates that are going to react with each building block we use. And then, of course, using some enzyme like T4 DNA ligase in order to do the ligation. We complete the second cycle and we pull everything together. If this is a 2-cycle library, we stop there. If we need to add more cycles, we continue with the same way. So in between the steps now, the synthetic, the chemical synthesis, and the encoding, we need to do some purification. And what we usually do is precipitation. So we take the nucleic acid solution, and by adding sodium chloride and ethanol, we can generate the salt of the DNA. And we centrifuge it, it precipitates, and then we discard the supernatant, and then we can reconstitute it in the buffer of choice for the next step. And ideally, by the end of every cycle, we would like to do a gel filtration using a size exclusion chromatography. And by doing that, we make sure that the big DNA conjugates are the only thing that goes through the column, and any small molecule like excessive reagents or DNA tag precursor remain in the column, and they don't interact with the next step. So for the affinity-based selections, as I mentioned before, this can happen in just one experiment. So we take the library as a mixture, and the first step is the incubation of the library with the immobilized target, which is followed by the removal of the non-binders. This is just a washing step, and then the next step is to denaturate the protein, elute the binders, which undergo sequencing in order to understand which small molecules are binding to the active site of the target. The big difference with high throughput screening, fragment screening, and other screening techniques is that we need to add an extra step after the affinity-based selections. And this usually happens because we need to do something that is called heat validation. So we need to synthesize these small molecules, these binders, without the DNA tag right now, and test these again, and in the same assays to make sure that the small molecule is indeed the one that is binding to the active site. And this is happening because the DNA is quite is bigger compared to the small molecule. And sometimes it can interact and create false positive results during the screening campaign. So this is something to have in mind. And also, most of the times we need to do some adjustments in the structure of the molecule because the DNA tag is hydrophilic, so this means that increases the solubility, and this most of the times is not happening with the small molecules based on the chemotype. So we need to do some modifications in the part of the small molecule that the DNA was attached before. And what we see here in the scheme is the difference between the original del hits and how these are developed during the medchem process. Some of them can be quite similar. As you can see here, we just have a change of oxazole to triazole, but some others can be quite different. Here we're having a whole amine missing, and of course here the substitution pattern from 1,4 goes to 1,3, which gives completely different chemistry to the molecule. So in our lab and in an academic environment, we can use DNA-encoded libraries in two different ways. The first one is to use commercially available libraries that are provided by vendors like XChem, Lucy Aptec, HitGen, and many pharmaceutical companies. And we can have access to huge libraries of billions of compounds. And we purchase this library, we do the affinity-based selections in-house, and then we give the data back to the company for the sequencing and the hit reporting. And the big drawback with this way of working is that we have no idea about the library composition, no idea about the chemotypes that are included in the library, and if these can be compatible with the target we are working on. So this is the easy way, I would say, to do it, and it's really preferred by academic groups nowadays because you don't have to spend any time in order to design a library, optimize the reaction conditions, and synthesize the actual library. You just go to a vendor and you can buy a library, which is really handy if you want to do some quick experiments and see if you have some hits for your target. We've been doing work in this way, and hopefully soon we'll be able to share some results for some hits regarding LPX8. But today I will talk you more about in-house synthesized libraries, which is something else we are also doing. The difference here is that in order to synthesize a library in-house, you need to have a specific infrastructure. And of course, you need to have some expertise, knowledge on how to synthesize a library. And of course, by doing that, you can have full control regarding the design of the library and how diverse the building blocks you are going to choose. And this has as a result the diversity of the final components in the library. And of course, by following this procedure and doing an in-house library, you can definitely create target-focused libraries. For example, let's say we are having already a known inhibitor, but he has a specific pharmacophore that we know that is tolerated. So we keep this pharmacophore and then we change other parts of the molecule by just synthesizing a library. So by doing this, we can see if we can pick up new pharmacophores with that are compatible with the target structure. So we wanted to synthesize a library like this, not a targeted. We went for a general purpose library, but we want to synthesize an in-house library. And we just went through our chemical storage areas. And we wanted to synthesize a library that can be done by using one type of reactions. And as you saw in the title of this presentation, we are using amide conformation. And we wanted to keep the cost as low as possible. So most of chemical labs are having quite a few amino acids and carboxylic acids already in stock. So by keeping the cost of the building blocks quite low, by using things that we already have, that was a very good starting point. So we went through our database and we managed to find 65 FMOC amino acids and 127 carboxylic acids that had really interesting structures. And then after having a conversation with colleagues that are doing computational chemistry, they said that we can try and optimize this selection of building blocks. So they wanted to do some computational studies to see if they can maximize the diversity, and this can probably lead to a better binding potential to the target. So what they did was that they gathered almost 7,000 commercially available amino acids. And by using Schrödinger QuickProg, which is a computational chemistry program, they generated all the molecular features for these amino acids. And molecular features like molecular weight, lipophilicity, hydrogen bond donors and acceptors, things that medicinal chemists usually look when they look at a molecule. And they generated these features and they just plotted all these characteristics in the graph that you can see to the right of this slide. And so they mapped the diversity of the amino acids in this plot. And in this graph with orange, you can see the amino acids used in the original design of the library, and with blue, all these 6,961 amino acids that were screened. And we tried to identify underrepresented areas of in this plot. And I mean, these amino acids, most of them are quite exotic, I would say. You can imagine that in the side chain of amino acid, you can install whatever you want. So with an exotic structure also comes an exotic price. And since we wanted to keep the cost as low as possible to showcase that this is something that if you have the infrastructure and the knowledge, you can do it without spending a huge amount of money, we decided to buy 7 additional amino acids, which are the ones represented with green in the graph. Some of them are quite close to amino acids of the original design, but some others are quite far, and this is going to give some diversity in our library. So in this scheme, you can see the properties of the compounds of the original dataset to the left and of the modified dataset to the right. And what we can mostly distinguish is that the compounds in the modified dataset of low molecular weight are having higher lipophilicity compared to the ones in the original dataset. But what we can easily distinguish is that we have increased the number of compounds in this area with higher molecular weight and higher polar surface area, which is really good. But unfortunately, the last step before starting the actual synthesis of the library is something we call building block validation. So after optimizing the conditions we are going to use for the library, we need to validate the building blocks. If you remembered earlier when I was talking about DNA-compatible chemistry, I mentioned that we need to have reactions that can give high yield in order not to have too many byproducts during the reactions. So we don't have truncates by the end, and this makes the mixture complicated and the sequencing as well. So we validated with 72 amino acids, and 60 of them were high yielding, so they were included in the library. And we did the same with the carboxylic acids. From 127, we tested representative examples, and we decided to include 112 of them. So for the final synthesis of the library, of course, as I mentioned before, we start with a DNA headpiece, and this library consists of 3 cycles. In the first and second cycle, we introduce amino acids, and on the third cycle, we cap the sequence with 112 different carboxylic acids. And right now, this library is synthesized, and pretty soon we are going to do some selections experiment against some LPXH variants, but also some other targets that usually are used as benchmarks, like carbonic anhydrases and soluble epoxide hydrolases and so on. So the take-home message is that if you have the infrastructure and the expertise, you can generate DNA-encoded library with containing a significant amount of compounds. This library has 400,000 unique linear peptide-like compounds that can be screened. And yes, targets is one, as I mentioned before, is a general purpose. So it's not synthesized based on any known inhibitors. It can be used and screened against any target of interest. And of course, it can act as a very good starting point at early drug discovery programs. So at that point, I would like to thank everyone who has contributed to this project, my supervisors and my collaborators, and all of you for tuning in today.
Thank you so much, Chris. Otilia.
Thank you, Eva.
Now the Zoom has been updated and now the applauses go through the... That's nice. Oh, wow. This is a whole show. Thank you, everybody, for your applauses and for your attention. So now we have a little bit of time for Q&A before we take a break. And some of you might be thinking about your questions to Chris. We also listened to most of short versions of presentations by you guys at the retreat. So probably people have it a little bit recent also, talking about the DELs. Let me see if anybody puts their hand up. If not, you can also write your questions in the chat and I will read them for Chris. But maybe one question that we can ask is, so once you make your library, right? Like, do you directly use it after synthesizing it, or do you work with it in a way to validate it first and then you use it like you want to try to find new combinations of molecules?
Oh, can you repeat? I don't think that I fully understand.
Okay, so once you finish to synthesize your library,
yeah,
can you use it directly, or do you have to check things after it's synthesized before you can actually use it?
Use it for affinity-based selections.
Yeah.
The only thing you need to do is to do some PCR to make sure that the DNA barcode is intact and you can amplify it. But after this is successful, you can continue with selections.
Do you do so the sequencing, because I come from the background of molecular biology and genetics, and we do a lot of sequencing.
Yeah.
Do the sequencing, and you were saying, for example, when you are buying your library, it's already done. It's easy because it's a standardized thing, and the company that sells you that, it also sends you kind of the analysis. In this version, are you the one that takes care over the whole workflow of also doing the sequencing and checking the sequences and checking that everything is correct with the sequencing?
I mean, not personally. I mean, up to now, I've been part of the design, optimization, and synthesis. I'll take part in the selections as well. But then we will send the data. We are collaborating with SciLife Lab in this project, so people in SciLife Lab are going to take care of the sequencing and the data analysis.
Okay, so then you basically, what you get, you are not really involved in that part of the sequencing and
No.
basically get the results on the other end. I was just thinking if you also had to, learn.
No, I think my part will stop at the selections.
You don't look at chromatograms and trying to see how.
No. I have no idea how it is done, to tell you the truth.
All right. We'll ask your thesis opponent not to ask you anything about sequencing then.
I mean, I would know the basics, but it's definitely not my expertise.
Yeah, I understand. It has to be somewhere, and that's the idea with collaborators and having to work with different expertises. But it's a lot of the different steps that you are doing yourself, so it's really great.
Yeah, it's quite of interdisciplinary projects with learning new stuff. It's really nice.
Great. Does anybody have a question regarding the DELs? Here we had the Ellen. All right. If nobody has any questions, of course, I know Chris is open on the mailbox if anybody wants to either ask a question or perhaps they think that this kind of technology could be used in their projects, or some of the knowledge that comes from this technology could also be explored for the projects. That's what we're trying at the UAC, that people from different disciplines get to understand and maybe instill some of that into their own projects. So I'm sure that you'll be happy to get those questions.
Definitely. Feel free to bother me.
Nice. So with that, we will take now about 5-minute break. So it's 7 past 8 past.